Bismillahirrahmanirrahim, and welcome back to this next video. And from this video, we are going to start our discussion on the nucleic acids. Now, these nucleic acids, they usually come in two varieties, the deoxyribonucleic acid and the ribonucleic acid. So we are going to have a detailed discussion on both of the deoxyribonucleic acid and the ribonucleic acid. So the first thing is, what are nucleic acids? These nucleic acids, sometimes also known as the polynucleotides, they are biopolymers present in all living forms, and they are very essential for the different functioning in the living organisms. So, before going into the details on the nucleic acids, why we call them is the nucleic acids. So, this term nucleic acid, it have got a historical perspective. If you look at the discovery, so the nucleic acid, they were discovered in the nucleus of the white blood cell in 1869 by the Swiss biochemist Miescher. And at that particular time, it was not fitting into the two major classes of the biomolecules, the proteins or the lipids. So he called them as the nuclein because it was not fitting into the protein or the lipid category. So if you look at these nucleic acids, they are acidic because they can donate hydrogen ions to the solution, giving them acidic characteristics. And this acidic characteristic is due to the presence of the phosphate group in the backbone of the nucleic acid that we will see in detail. So if you look at this name, the nucleic acid, so it combines their discovery location. It was discovered in the nucleus, and their chemical property, they are acidic because of the presence of the phosphate group. So as they are present in the nucleus, they have got this acidic property, so we call them is the nucleic acids, the acids that are present in the nucleus of the cell. If you talk about the types of the nucleic acids, with respect to the molecular biology, we classify them into two major types. One is known as the deoxyribonucleic acid, and the other one is known as the ribonucleic acid. Now, both of these nucleic acids, the DNA and the RNA, their building blocks is known as the nucleotides, and the nucleotide is composed of three components. The first component of the nucleotide, that is a pentose sugar, which is actually a 5-carbon sugar. Then you have got a phosphate group, and then you have got a nitrogenous base. So the nitrogenous base plus the phosphate group plus the carbon, we call them is the nucleotides, and these nucleotides, they are the building blocks of the DNA and the RNA. So let us have an image on the nucleotides. So if you look at over here, this one is your sugar, this pentose sugar, this one is your nitrogenous base. It can be any of them, as I'll tell you about the different type of the nitrogenous bases they are there in the nucleic acid. And then we have got a phosphate group. So the phosphate group plus the pentose sugar plus the nitrogenous base, we call them is the nucleotide. Now there is another term that is used, which is known as the nucleoside. So these two terms that should not be confused, because the nucleoside and the nucleotides, they are different from each other. Now when the nitrogenous base combines with the sugar, this particular structure is known as the nucleoside. But when we add a phosphate group to the nucleoside, it becomes the nucleotide. So there are three components in the nucleotide: the phosphate group, the sugar, and the base. But in the nucleoside, this phosphate group is absent, and we only have the sugar and the base, and we call them is the nucleosides. And of course, there are a variety of the nucleosides, and we will discuss them in detail during this particular course. So let us start about the different components of the nucleotide. So if you talk about the components of the nucleotide, as I've told you, there are three. So we are going to focus on the first one, which are known as the nitrogenous bases. Now, if you look at the literature, or if you look at the books, you are going to find three terms. Sometimes they are using the nitrogenous base term, sometimes they are using the nucleobase, and sometimes only bases they are used. But these three things, the bases, the nucleobase, or the nitrogenous base, they refer to one in the same thing. So these nitrogenous bases, they are actually nitrogen containing biological compounds, and they are the component of the nucleotides. And I've told you that these nucleotides, they are actually the building blocks of the nucleic acids. So we are dealing with the nitrogen containing compounds when you talk about the nitrogenous bases. Now these nitrogenous bases, they come in two flavors. One, they are known as the purines. Now these purines, they are two ring structure consisting of a fused imidazole and pyrimidine ring. If you look at this particular image, this particular ring structure, this is known as the pyrimidine. This particular ring structure is known as the imidazole. So we are dealing with a two ring structure. So one of the ring, that is the imidazole ring, and the other one, that is the pyrimidine ring, and they are fused to each other. So this is one type of the nitrogenous base. The second type of the nitrogenous base, they are known as the pyrimidines, and these pyrimidines, they are a single ring structure. So if you look at their structure, so their structure is a single ring. What that means is that this is a smaller molecule in comparison to the purines, because they have got two rings over here. So let us first talk about the purines, that which kind of the purines they are there in the nucleic acids. So the first important purine that is present in the nucleic acid, that is known as the adenine. And this particular image is showing you the standard structure of the adenine with standard numbering of the position. So the numbering starts from here, from this particular nitrogen, the first nitrogen of the pyrimidine ring, and it's gone on like 1, 2, 3, 4, 5, 6. So there are 6 carbons in the pyrimidine, and then you have got the 3 over here, like the 7, 8, and 9. So this is the standard numbering of the positions in the adenine. If you talk about the another purine, that is known as the guanine, and this particular image is again showing you the structure of the guanine with standard numbering of the position. So again, the numbering starts from this particular nitrogen, and it moves on until it reach the position number 9. So if you look at the purines, both of them, the adenine and the guanine, they have got this two ring structure. Now, these adenine and the guanine, they are present in both the DNA and the RNA. As I've told you that the nucleic acid have got two types, the DNA and the RNA. So the adenine and guanine, they are present both in the DNA and in the RNA. The second type of the nitrogenous bases, they are known as the pyrimidines, as I've told you. And there are three pyrimidines when you talk about the nucleic acid. The first one, that is known as the cytosine, and this is the structure of the cytosine. And the cytosine is present both in DNA and the RNA. The second important pyrimidine, that is known as the thymine, and this is the structure of the thymine over here. This thymine is only present in the DNA, and it is not present in the RNA. When you talk about the third pyrimidine, that is known as the uracil, and the uracil is only present in the RNA, and that is not present in the DNA. So when you talk about the nitrogenous bases, you can say that the adenine, guanine, cytosine, and thymine, they are the four bases found in the DNA, and the adenine, guanine, cytosine, and uracil are the four bases found in the RNA. So if you look at the difference between the DNA and the RNA with respect to the nitrogenous bases, the only difference is that the DNA have got thymine, and the RNA, they have got the uracil, and the other three nitrogenous bases, they are the same in the DNA and the RNA. Secondly, the structure of the uracil and the thymine, if you look at this image, they are very much similar to each other. The only difference being the presence of the methyl group in the thymine and the absence of the methyl group in the uracil. And I'll have a detailed discussion on that why the thymine is present in the DNA and why the uracil is not present in the DNA, and vice versa. The second component of the nucleotide, that is the ribose, which is the sugar component. If you talk about the sugar component, so a ribose, that is a simple sugar, and the naturally occurring form, that is known as the D-ribose, and this D-ribose is a component of the RNA, specialized type of the nucleic acid. Now this ribose, it have got structural analog which is known as the 2' deoxyribose, and this 2' deoxyribose is an essential component of the DNA. What that means is that the ribose is present in the RNA, the 2' deoxyribose that is present in the DNA. So the sugar component that is different in the DNA and that is different in the RNA. If you look at the structure of the two, so this one is the structure of the ribose, and if you look at it here at position number 2, you are seeing this hydroxyl group over here. Now this particular hydroxyl group that is not present in the deoxyribose, because the oxygen that is not present over here, if you can see at the position number 2 of the deoxyribose sugar, there is no hydroxyl group. At position number 2 of the ribose sugar, there is a hydroxyl group. So as I've told you, we call this as the 2' deoxyribose. The reason is because at the position number 2 of the sugar, there is no oxygen present. So therefore, we are using this term deoxy over here. Now, this nucleoside, if you can see over here, this would be just one of the example that the nucleoside consists of a nucleobase and a 5 carbon sugar. So when you talk about the sugar in the nucleoside of the RNA, that would be a ribose sugar. If you talk about the nucleoside of the DNA, that would contain the 2' deoxyribose. Another important thing to keep in mind is that if you look at over here, at every carbon position for its numbering, we are putting a prime over here. Like this is 1', this is 2', this is 3'. So the importance of this prime over here is that you can easily differentiate between the carbons of the sugar and the carbons of the nitrogenous bases, because you are not going to see any prime on the carbons of the nitrogenous bases. So this would be the differentiating thing between the sugar and the you can say the nitrogenous bases. The third component of the nucleotide, that would be the phosphate group. If you look at it here, this one is the whole nucleotide, because you have got the nitrogenous base, you have got the sugar, and you have got the phosphate group. So this one is the nucleotide, and that would be the building block of the DNA as well as the RNA. Now, why I'm showing you this particular figure, I want to focus on the attachment positions, because if you look at over here, now this in the nucleotide, this particular sugar, it is occupying a central position. Now the base is attached to the 1' of the sugar, as you can see over here, and the phosphate group that is attached to the 5' carbon of the sugar. So the sugar is actually the central position; it's occupying the central position, and to 1', the nitrogenous base is attached. That can be any of the nitrogenous base that we just discussed, and at the 5' carbon, there would be a phosphate group when you talk about the nucleotide, but you can attach extra phosphate groups over here. Like you can have 2 of them, you can have 3 phosphate groups. Say for example, if you talk about the ATP, which is the adenosine triphosphate, there at that particular in that particular ATP, you have got 3 phosphate groups. So this particular image is showing you the 3 components of the nucleotide, as well as the attachment position of the nitrogenous bases and the phosphate group to the sugar. And the sugar can be, of course, deoxyribose sugar. If you talk about the DNA, it can be a ribose sugar. If you talk about the RNA, but this particular image is showing that this is the nucleotide for DNA, because at position number 2, you do not have any hydroxyl group over here. So we are going to continue our discussion on the in the next video on the other aspects of the DNA and RNA.